Hello dear students, and welcome back to our chapter number 18, body fluids and circulation. So today we are going to learn about various types of blood groups which is present in our body. Okay, so let's talk about the blood group. So experiments with blood transfusions have been carried out 400 of the years. So many patients have died, and it was not until 1901 when the Australian Carl Landstein discovered human blood groups that blood transfusions become safer. Okay, so he found that mixing bloods from two individuals can lead to blood clumping. Okay, that is means a different reason. Okay, because the two individuals blood get clumped, so it will be harmful. So the clumped RBCs can crack and cause toxic reaction. Okay, so this can be felt in Carl Landstein discovered that blood clumping was a immunological reaction which occurs when the receiver of the blood transfusion has antibodies against the donor blood cells. Okay, again then Carl Landstein work made it possible to determine blood type and thus pave the way of the blood transfusion to carry it out safely. For this discovery, he was awarded the Nobel Prize in Physiological or Medicine in 1930. Okay, so the difference in human blood are due to the presence or absence of certain protein molecules that is called antigen and antibodies. Okay, so the antigens are located on the surface of the RBCs and the antibodies are in the blood plasma. Clear? So individual have different type of combinations of their molecules. Okay, so the blood group of you belongs to depend on what you have the inherit from your parents, because parents we having the inherited characters which came from our parents, so that will decide our blood group. Okay, so there are more than 20 genetically determined blood group system known today. So the ABO and rhesus, yes, that is known as the RH system, are the most important one which is used for the blood transfusions. Okay, not all body groups are compatible with each other. Mixing incompatibility bloods leads to blood clumping or agglutenations, which is dangerous for the individual. Okay, means blood clumping is very dangerous. Now let's see the blood groups. Okay, so see my dear students, here it is given a point, that is ABO blood group system. Okay, so in ABO blood group system, it is the typing system. There are 4 different kinds of blood types. Okay, that is A, B, B and O, that is called null. Okay, so blood group A. Okay, if you belongs to blood group A, you have A antigen on the surface of your RBCs and B antibodies in your plasma. Okay, if you having blood group B, then you have B antigen on the surface of the RBCs and A antibody in your plasma. Okay, but if you belongs to blood group AB, that means you have A and B antigen on your surface of the RBCs and AB antigen of all the body blood plasma. Clear? Now let's talk about blood group O. Okay, so if you belongs to the blood group O, that is null, you have neither A nor B antigen on the surface of the RBCs, and but you have both antibodies A and B in your blood plasma. Okay, so here the table shows the 4 ABO phenotypes. See here. Yes, and that is known as the blood groups that presence in a human population and the genotype that give rises to them. Clear? Now blood group O, blood group A, blood group B and blood group AB. Clear? Now you have see here, it is a table given here. Now blood group, the antigen, the antibodies, and the genotypes. Okay, genotypes means the genetic constitution of an individual organism. Clear? Now let's talk about more why group A blood must never be given to group B person. Okay, so there is a question why we can't give. So giving someone blood from the wrong ABO group could be fault. Okay, means it will be harmful, and the antigen A bodies in a group B attack group A cell and vice versa, and blood group O negative is a different story. Okay, O negative is a different story. Clear? Now blood group, now we will see the blood group compatibility, means how we can compare different type of blood group. Now the table demonstrate blood type of compatibility. See here, this is the table. Okay, a person with AB positive blood type can receive blood from all the major blood types. Okay, means those who having AB positive, they can receive from anyone, means they are universal acceptor. Okay, and at the other extreme, people with O negative blood type can only receive blood from the donor with the same blood type, means those who having O negative, they can only receive with the same blood group person, means those person having the O negative. Clear? Now see here, the table is given, how beautifully it is given. So here is blood group. Now gives to these groups, means how we can give to them, to whom we can give, and to from where we can receive. Okay, yes. Now let's talk about the RHS, the RH blood group system. Okay, so must learn this table. Okay dear students, this is for your general knowledge and for information. So the RHS, that is RH blood group system. So blood type were discovered in 1940 by Carl Landstein and Alexander Wiener. The RH system was named after RHS term pronounced monkey, since they were initially used in the research to make the antiserum for typing blood samples. If the antiserum angiostinate your red cells, you are Rh positive. It does not, you are Rh negative. Clear? So the Rh factor is an inherited protein found in the RBCs. A person whose blood has this protein is Rh positive, and who lack this protein, that is Rh negative. Clear? Well, it's get more complicated here, because there's another antigen to be considered negative, the Rh antigen. Okay, so some of us have it, some of us does not. So if it is present, the blood is Rhd positive. If it is not, then it's Rhd negative. Clear? This means you can maybe one of either blood group, that is you can see here, yes, ARH positive, that is A positive, ARH negative, A negative, BRH positive, B positive, BRH negative, B negative. Okay, now what is the incompatibility in RH? So let's see about it. So the Rh incompatibility is a condition that develops when a pregnant woman has Rh negative blood and the baby in her womb has Rh positive blood. Okay, so Rh incompatibility develop only when the mother is Rh negative and the infant is Rh positive. Okay, that means the baby is positive but the mother having the negative. Okay, so during delivery the Rh antigen enters mother circulation through the breast in the placenta, and mother make anti-Rh antibodies during or after first pregnancy. Okay, and after mothers are anti-Rh antibodies that cross the placenta and destroy fetal blood cells. Clear? So, and some causes, yes, we can see. During pregnancy, RBCs from the unborn baby can cross into the mother's blood through the placenta. Okay, so if the mother is Rh negative, her immune system treats Rh positive and felt cells as if they were a foreign substance. Clear? So the mother's body make antibodies against the fetal blood cells. These antibodies may cross back through the placenta into the developing baby. Okay, and they destroy the baby's circulating RBCs. This result in the hemolytic disease of newborn. Yes, that is known as HDN, and this disease is called the erythroblastolysis fetiolysis. Okay, when red blood cells are broken down. Clear? Now let's talk about the circulatory system in human being. So, man is the most developed and highly evolved organism on the earth, right? Its circulatory system consists of two parts, that is blood vascular system and lymph circulatory system. So, the two systems collectively perform the function of circulation and distribution of the gases on the other material in the body tissues. So, body tissues, now blood circulatory system of man. Okay, so let's talk about it. This is the circulatory system in which medium, that is liquid of the circulation, is blood. Like other mammals, the blood circulatory system of man is constructed by blood, heart and blood vessels. So in human heart, that is appearance and position. Okay, so the mammalian heart, that including man's heart, is a cone shaped hollow muscular organ. You can see how beautiful it is. Yes, so it is a cone shaped hollow muscular organ which lies in the thoracic cavity. Yes, and mediastratum, that is known as the center, the space between the two pleural sacs, and the apex of the heart is slightly tilted down left. Okay, and it protected by the bones and muscles of the chest wall, ribs, backbone, breastbone and diaphragm. So human heart is about the size of first, measuring about 12 centimeter in the length and 9 centimeter in the breadth. Okay, and its weight is about 300 gram. Clear? Now it having the protective covering, I have told you already. So heart is an enclosed and is a transparent membranous bag that is called the pericardium, and it consists of two layers, that is called fibrous pericardium and serous pericardium. Fibrous means which is fulfilled with the different type of fibrous tissues, and serous means different types of serums, okay, which make it smooth and moist. Okay, so for the serous, we having two membrane, that is parietal layer and visceral layer, which we already know. Yes, which we already studied. So the function of pericardium protect heart from the injury and against the frictions, and pericardial fluid keep the heart moist and protect it from external shock and frictions at the time of the beating. Clear? Now below the pericardium, the dark brown color heart is located. It is surrounded by the thick wall which is called the heart wall. Inside this wall there is a heart cavity. Okay, and the heart wall consists of three layers, that is epicardium, mesocardium and endocardium. Okay, so the epicardium is the outermost layer of the heart wall which is made up of the connective tissue, and it protects heart from the external jaws. And mesocardium, it is the middle layer, consists of the cardiac muscle, and it has the feature of both voluntary and involuntary muscles. Okay, and it shows cardiac movement till the end of the body's life. Clear? Now endocardium means the innermost and thin and soft layer of the heart wall, and the cell of this layer from the cardiac muscles. Let's talk about the structure of human heart. So the chamber, first is chamber. So as we all know that we having four chambers, that is two auricle and two ventricle, right? So let's talk about the auricles or artery. Okay, so it is forms a smaller upper part of the heart. It is composed of two upper thin walled chamber of the heart, and it's separated by the inner inner ocular spectrum, which providing mixing of oxygenated and deoxygenated blood. Now, right atrium or auricle is large in size, and intra-auricular spectrum has an oval desperation that is called fossal ovulis, which is the vestigial of an ovule apparatus. Clear? Now, auricles are blood receiving chamber, and right auricle receive deoxygenated blood from the superior and inferior vena cava, and it is also receive blood from the coronary veins, and the left auricle receive oxygenated blood from the lung by 4 pulmonary veins, that is 2 from each lung. Clear? Now, ventricles. Ventricles are the ventricles are the two thick wall lower chamber of the heart. Left ventricle is much thicker than right ventricle because it has to push the blood in all the body parts, while right ventricle has to push the blood to closely lying lungs only. Okay, so both the ventricles are spread by a thick obligated place, that interventricle spectrum, and ventricles are blood distributing chamber, that is left ventricle supplied blood to the body by a single aorta, whereas right ventricle supplied blood to the lung through pulmonary aorta. Clear? Now let's talk about the opening, the walls and the opening. Okay, so first we having is the cuspid walls. So the entry ventricular opening between the left atrium and left ventricle is guarded by the bicuspid valve, that is also known as the mitral valve. Okay, so it having the two flaps. The right entry ventricle opening is graduated by the tricuspid valves, has the 3 flap, means in the back, it is present on the back side, and next we having is semilunar valves. Semilunar valves means this is the half moon shaped flap arising from the base of the pulmonary trunk and aorta. Okay, now cardiac wall. So what is cardiac wall? Histologically, the wall of the heart is formed the following 3 layers, that is visceral pericardium, myocardium, and endocardium. Okay, so visceral cardium, it is the outermost layer of the heart wall, which is formed by the Coleman epithelium of flat cells, and myocardium, it is the middle layer formed of cardiac muscle fiber, and endocardium, it is the innermost layer, consists of simple squamous epithelial cell. Clear? So all these things you can see in this structure. Okay, and you must learn how to draw this structure. Clear? Now we having various paths for the blood flows, right? So that we will discuss in our next class. Till here we will end our video, and rest we will do in our next class. Thank you.